Hello everyone, welcome back to our channel. I'm Dr. Ashmita. Today in this video, we'll study about the receptor and signaling pathways. In my introduction video, I have said about the 6 kinds of receptor and their signaling pathways. Now we'll study the first receptor tyrosine kinase. So this is the image taken from Robin's textbook. Here these are the cell membrane receptors. Here we are going to study about this tyrosine kinase receptor. So these receptor tyrosine kinases, they belong to a class of cell surface receptors. So here this is a receptor present on the cell surface. This is present on the membrane. So they belong to a class of cell surface receptors with intrinsic enzymatic activity. Intrinsic enzymatic activity because they have this kinase domain. Kinases means it is an enzyme. Kinase is an enzyme. It will add phosphate group. It will add phosphate group to whichever molecules they come in interaction with. So we are calling it a receptor tyrosine kinase. Therefore, these receptors have inbuilt, intrinsic means inbuilt enzymatic activity. That enzymatic activity is specifically tyrosine kinase activity. Therefore, the receptor is named as receptor tyrosine kinase. Receptor with a kinase activity, that means it will add phosphate to the tyrosine. Therefore, it is called a tyrosine kinase, receptor tyrosine kinase. And this receptor tyrosine kinase, they play a central role in signal transduction. Obviously, we are studying about transduction pathways. So, this receptor tyrosine kinase play a very important role in signal transduction that is responsible for cell proliferation. Number one, cell differentiation, metabolism, and survival. So these pathways help in these functions: proliferation, differentiation, metabolism, and survival. Let's see the structure of a receptor tyrosine kinase. So this image from the Robbins, here this is the tyrosine kinase receptor. These are the monomers. This is one receptor, this is one receptor. So the structure of tyrosine kinase receptor, it has an extracellular domain. Extracellular means which is present outside the membrane. This is the membrane. This surface, this part of the receptor present outside the cell membrane is called extracellular domain. This is an extracellular domain. And the part of receptor present within the membrane, which is embedded within this membrane, within the span of this membrane, is called a transmembrane. It is a transmembrane domain. And part which is cell, inside the cell within the membrane, that is called intracellular or cytosolic. Since the part of this receptor present, it is floating in the cytoplasm, therefore it is also called a cytosolic. It is cytosolic domain. So this receptor has 3 parts: extracellular domain, transmembrane domain, and the intracellular or the cytosolic domain. This extracellular domain, it will act like an antenna. Antenna means it will receive the signal. So this extracellular signal, it is the extracellular domain is the first part to receive that signal. So it will sense and detect the extracellular signals like the growth factors, hormones or cytokines. Whatever signal is coming from the external environment is first sensed, it is first perceived by this extracellular part of the receptor, and it will ensure that the receptor will respond only to the specific signals. That means here the example is a growth factor. Growth factor is a ligand. This receptor is a growth factor receptor. It will ensure, this part will ensure that it is responding only to its specific ligand. If it was an epidermal growth factor receptor, it will respond to only epidermal growth factor. If it was a vascular endothelial growth factor receptor, it will respond only to the vascular endothelial growth factor. So it is highly specific. This specificity, this precision is brought about by this extracellular domain. And this transmembrane domain, it acts like a shaft. It is like a mechanical anchor or stability. It will transmit conformational change. What is a conformational change? After ligand binds to this extracellular domain, the three-dimensional structure of this receptor will change. Conformational change means three-dimensional structure will change, and this change is transmitted from this extracellular part to the intracellular. From extracellular, from this extracellular, it has to be transmitted to the intracellular domain. It is transmitted via this shaft. It will like a mechanical anchor. It will transmit a conformational change from this extracellular part to the intracellular part upon this ligand binding, and it provides structural stability. That means whenever a receptor gets activated after ligand, the two monomers will come together, form a pair. So this monomers are aligned during dimerization. This is called dimerization during the pairing up. The receptors are aligned. That means they are like parallel to each other because of this long transmembrane shaft. This is the function of a transmembrane domain. It will first of all transmit the signal from extracellular to the intracellular, and also keep the two monomers aligned during dimerization. Coming to the third part, that is the intracellular or the cytoplasmic tyrosine kinase domain. This intracellular part of the receptor has this main component, that is the enzyme activity. What you are calling is this kinase domain. The kinase domain is the main machinery or the engine of the receptor. This domain has tyrosine kinase activity, means it will add phosphate. It will here this tyrosine kinase domain is present. The kinase domain present in the intracellular part will add kinases. It will add phosphate groups to the molecules it will come in contact with. It will add the phosphate from the ATP. From ATP, it will recruit phosphate, and this phosphate will be transmitted to the tyrosine residues of the self receptors of it present in its own self, or to the other receptors containing tyrosine. So this is the function of intracellular domain. So the structure of receptor tyrosine kinase, extracellular domain which will act like an antenna, that means it will receive the signal and it will respond to it. It will make sure that it will respond to its only specific signals. Specificity of the receptors brought about by extracellular domain. Transmembrane domain. That means it will transmit the conformational change. It will transmit the signal from extracellular to the intracellular domain, and also gives a mechanical stability, a structural stability during dimerization. Intracellular part, it is the main engine or the machinery containing kinase. It will have this kinase enzyme. Kinase means it will add phosphate to what? To the tyrosine of the self receptor, to itself or the other receptors. This is the structure, basic structure of a receptor tyrosine kinase. Let's move into the main mechanism of how this receptor tyrosine kinases act. The story begins with ligand binding. First, ligand has to bind. So, ligand binding. Here, this is the growth factor. It will bind to a growth factor receptor. After ligand binding, there is receptor dimerization. That means the two monomers, this is one monomer, this is one monomer, they come together, they form a couple or pairing up. That is called dimerization. After dimerization, autophosphorylation occurs. That means the kinase domains will phosphorylate itself. It will phosphorylate that own receptors. That is autophosphorylation. After autophosphorylation, there is recruitment of adapter and effector proteins. Once this got autophosphorylated, other adapter protein, this is an adapter protein here. This got recruited, and many other effector proteins get recruited towards this activated receptors, like the PI3K get recruited, AKT get recruited. In that way, the activated receptor will recruit adapter effector proteins. Lately, there is activation of downstream cascade. Because of this activated receptor, it will in turn activate other downstream, like RAS, from membrane towards the interior of the cell. That is a downstream cascade. From here, it will move towards the interior of the cell. A downstream cascade chain of reactions occur from the membrane towards the interior of the cell. So, let's discuss in detail. We'll begin with ligand binding. After ligand binding, for example, a growth factor bound to a growth factor receptor. Because of ligand binding, there is conformational change, that is three-dimensional structure. 3D structure of the receptor will be changed. 3D structure will change. It will change in such a way that the dimerization surface get exposed. That means the attachment, dimerization, coupling, that surface gets exposed. Because of this conformational change or the three-dimensional change, there will be receptor dimerization. This is one monomer, this is one monomer. They come together, they form a pair, a couple, they form a couple. That is pairing up, receptor pairing up, pair up of monomers, two monomers pair up. That is dimerization. And it will dimerize in such a way that kinase domains are juxtaposed. Here the intracellular part have this kinase domain. They are juxtaposed. That means they are just adjacent to one another and they transphosphorylate. Transphosphorylate means this is one monomer. Consider this is the first monomer, this is the second monomer. This first monomer will add this kinase of the first monomer will add phosphate to the second monomer and the second monomer will add phosphate to the first one. That means they are phosphorylating each other. This will add to this one exchange. The second will add to the first one. You give me one, I'll give you one. Like that. It's like a friendship. It's like a because they are couples, they are forming a pair. It will give to the other and will receive from it also. This is called transphosphorylation or autophosphorylation because they have coupled up, they are phosphorylating each other. It's like a self-phosphorylation, hence also called autophosphorylation. We are saying it is phosphorylating. What is it? Phosphorylating. What is the kinase domain? Phosphorylating. Tyrosine residues. So these receptors, they have tyrosine residues, which is one of the amino acids. Which is one of the amino acids. So the receptors have amino acids. Amino acids, one component is this tyrosine residue. Tyrosine residues are present. This kinase will add phosphate to the tyrosine residue. So the phosphotyrosine is formed. Whatever the byproduct came out, it is a phosphotyrosine. That means a tyrosine with added phosphate. That is phosphotyrosine. From here, what you can infer is phosphotyrosine is present in activated receptors. Obviously, because of ligand binding structure change, they both paired up, they transphosphorylate, they added phosphate to each other. Adding phosphate to tyrosine, that is phosphotyrosine, is ultimately formed phosphotyrosine. Phosphotyrosine is therefore present in activated receptor. Now, this phosphotyrosine, they create SH2 or the PTP domain binding site. Whatever that means whatever the phosphotyrosine is formed here, they will act as a binding site. They act as a binding site for SH2 or the PTP domains. What are these SH2 or PTP domains? These SH2 or PTP domains are present in other molecules, other downstream cascade like PI3K has it, AKT has it. Here we'll see this cycle, this flow. This PI3K molecule present inside the cell or the AKT molecule present inside the cell, they have this SH2 domains, SH2 side, SH2 domains are present, SH2 molecules are present. These get recruited towards, they get attracted towards this binding site. Therefore, the activated receptors will recruit the intracellular signaling molecules. So the phosphotyrosine, they create SH2-PTP domain binding sites. They create the domain binding sites. The domains are present in other molecules. So they create this domain binding site for downstream signaling protein. The downstream signaling proteins are this PI3K, AKT. We are calling downstream because they're present from the membrane, they are going down towards the interior. So this is about the phosphotyrosine. After this, what will happen? There will be recruitment of adaptor and effector proteins. The phosphotyrosine has been formed. Phosphotyrosine has this binding site. So this will attract an adapter protein. It will recruit an adapter protein. It will attach to the SH2 or PTP binding site of the phosphotyrosine, and which in turn it will recruit exchange factors or the enzymes. So here This phosphotyrosine, it has recruited this adapter protein. This is the adapter protein. This adapter protein, in turn, it will recruit other exchange factors, like, for example, it will recruit GRB2. It has recruited GRB2. GRB2 in turn will recruit SOS. Remember, SOS. SOS, again it will recruit this inactive Ras. Inactive Ras, because we are calling it inactive because it is bound to GDP. GDP means guanine diphosphate. That means it has two phosphate groups. This SOS, since it is bound to this phosphotyrosine, that means it is recruited from the phosphotyrosine. Phosphotyrosine recruited GRB2, it will give off its phosphate. This in turn will recruit SOS, it will give off its phosphate. Now this SOS, it will give its phosphate to the inactive RAS, it will donate to the inactive RAS. So there is addition of one phosphate by this SOS. That means it will phosphate is now bound to GDP. Now it has become a triphosphate. That means GDP will become the GTP because of addition of phosphate. This triphosphate. Here, inactive RAS bound to GDP with two phosphates. SOS has come donated its phosphate to the inactive RAS. That means the GDP will now become a GTP, triphosphate, addition of other phosphate. Now this RAS, which is bound to the GTP, it is called an active RAS. Now it has become active. This is called the active RAS. This is how the activated receptor has caused the activation of RAS. Let's summarize again. Because there is phosphotyrosine, it has recruited GRB, it has recruited adapter protein recruited GRB2 from their SOS. SOS will recruit the inactive RAS which is in bound form to GDP, that means 2-phosphate. This SOS will transfer its phosphate to the inactive RAS. That means 2-phosphate plus one additional phosphate. It has become triple. This is called GTP. GTP bound form is called active RAS. This is how the activation of RAS has occurred because of activation of this receptor. So it has caused RAS activation. Later, activation of downstream cascade. Just like how it has activated RAS, it can also activate other downstream cascades. So this receptor activates several branched kinases cascades and propagate and amplify the signal. The principal branches that we studied are this RAS, just now have said RAS pathway, Ras Raf MEK ERK. We'll discuss in detail now. Other branch is this PI3K pathway, PI3K. One branch is RAS, other branch is PI3K AKT mTOR pathway. We'll study each of these branches in detail. First, let's see the RAS pathway, RAS and RAF pathway. I have discussed till we have known how active RAS has formed. From the active RAS, that means activated RAS now will recruit an activated RAF. RAF. RAF is present in the cytoplasm, which is the full form is rapidly accelerating fibrosarcoma. Rapidly accelerating fibrosarcoma is the full form of RAF. RAF is recruited by this activated RAS. I forgot to tell about RAS. RAS is rat sarcoma family. Rat sarcoma family is the full form of RAS. This active RAS will now recruit and activate the RAF, rapidly accelerating fibrosarcoma. This RAF is a triple kinase. That means it has three kinases, three enzymes. MAP, that is MAP, kinase. MAP means the full form of MAP is mitogen activated protein. Mitogen activated protein kinase. The triple kinase is this RAF. This activated RAF, again it will phosphorylate. It will add phosphate to MEK. MEK. It will add phosphate to the MEK. MEK is a double kinase here. Double kinase. Again it is a mitogen activated protein kinase. This activated MEK, it will again in turn activate, that it will in turn phosphorylate and activate ERK. ERK. ERK means, again, it's a single kinase, mitogen activated protein kinase. So, to summarize, this active RAS thus formed, the active RAS thus formed because of this phosphotyrosine, it will recruit, the active RAS will recruit RAF. RAF is a triple kinase, mitogen activated protein kinase, kinase, kinase. This RAF in turn will phosphorylate. In each step, there's a phosphorylation. It will in turn phosphorylate MEK. It will phosphorylate and activate MEK, which is a double kinase, mitogen activated kinase, kinase. Again, it will phosphorylate and activate single kinase. Here is the ERK. ERK is called mitogen activated protein kinase. In each step, there is phosphorylation involved, and from triple kinase, it is becoming double kinase, then single kinase. Now, finally, here single kinase, that is mitogen activated protein kinase, will get translocated to nucleus. It will enter the nucleus here. This is the nucleus. It will enter the nucleus. Inside the nucleus, there are certain transcription factors. These are the transcription factors. Transcription factors. Transcription factors are also phosphorylated. This kinase will add phosphate to the transcription factors. Now the transcription factors got activated. It has become active. And activated transcription factors will cause expression of certain genes like MYC protein and also the cyclin D. These are the few genes expressed because of transcription factors. Cyclin D or the MYC protein, they are involved in cell cycle progression. They'll cause cell cycle to entry. They cause the cell to enter the cell cycle and cause proliferation. Therefore, ultimately cell cycle progression occurs. Cyclin D, we have known from this image also taken from Robbins. This is the cell cycle from G1 to S. All the phases of cell cycle are present. In the first phase, from the transition of G1 to S, the cyclin D plays an important role. It's the starter. The starting cycle, cyclin is the cyclin D involved in cell cycle progression from G1 to S phase. So this is how this Ras Raf pathway occurs. The Ras Raf pathway we have studied how it occurs in steps. So now we'll study about the second pathway, that is the PI3K-AKT mTOR pathway. Here it begins with activation of PI3K. PI3K is called phosphoinositide phosphoinositide 3 kinase. Phosphoinositide 3 kinase. So here begins with PI3K, that is the phosphoinositide 3 kinase. This PI3K has two monomers, like it is a heterodimer composed of P85 and P110. It has two molecules, P85, P110. This P85, it has SH2 domain. I have said in the beginning, the phosphotyrosine is a binding site for SH2 domains. SH2 domains present in PI3K. So one of the component of PI3K, P85, has this SH2, and P10, it will act as a lipid kinase. This P85 with SH2 domain, it will get recruited, it will come towards the membrane and bind to phosphotyrosine or the receptor tyrosine kinase. Here, this P85 has come towards the cell membrane bound to this phosphotyrosine. This P85 in turn, it will also activate P110. It will activate the P110. P85 with SH2 will bind to phosphotyrosine of the receptor tyrosine kinase, activate P110 catalytic subunit also. That means the entire PI3K has got shifted towards the cell membrane. This PI3K, it will phosphorylate certain membrane lipids like this PI3K has come here to the membrane. It will phosphorylate PIP2 into PIP3. So PI3K will convert the PIP2 present in the membrane into PIP3. This PIP3 will act as a second messenger. It will act as a lipid second messenger. Since it is present within the membrane, it acts as a lipid second messenger. Lipid second messenger means now it will recruit AKT. So from here to here, it is the first step. That is activation of PI3K. P85 will be brought towards this phosphotyrosine. It will in turn activate P110. The entire PI3K is brought here. It will convert PIP2 into PIP3, which will act as a second messenger. This is the first step. This PIP3 will now recruit AKT present in the cytoplasm. This recruitment of AKT to the plasma membrane, this AKT will now bind to PIP3. This AKT from here, it will go to the membrane, it will bind to PIP3. So PIP3 plus AKT is in bound form. This is now, it is said to be in active form. It is now activated. AKT is now activated. AKT is now activated. Once it gets activated, AKT, once it gets activated, now it will dissociate from the membrane. It is still, all these reactions are happening at the membrane level. Activated AKT will dissociate, it will detach from the membrane, come inside the cell. It will dissociate from the membrane. And in the interior of the cell, it will phosphorylate multiple cytoplasmic and nuclear substrates. Once it enters inside, this AKT will activate many cytoplasm. This is the cytoplasmic substrates or the nuclear substrates. It will further activate the other phosphorylate, activate other substrates. This is about step 2, recruitment of AKT to the plasma membrane and how it gets after activation, how it dissociates and come inside the interior and phosphorylate activate other cytoplasmic and the nuclear substrates. Coming to the step 3, that is, it begins from this activated AKT. This activated AKT, it has function of anti-apoptotic role and inhibit pro-apoptotic transcription. Anti-apoptotic means this AKT will phosphorylate and inactivate BAD. BAD means it is a pro-apoptotic protein. BAD is a pro-apoptotic. That means it will induce apoptosis. It will induce the cell death. It will induce the cell death. So this AKT, it will add phosphate. It will add the phosphate and inactivate the BAD. So not all the phosphorylations, whenever I'm saying here, it got phosphorylated, it got activated. In the Ras Raf pathway, in each step here, there is phosphate group addition. Phosphate group addition, because of phosphorylation, every step is getting activated. But here, this phosphorylation is not activation. Here it is the inactivation. AKT will phosphorylate this BAD, that is the pro-apoptotic. It is a pro-apoptotic protein. It got phosphorylated. Here it got inactivated. Inactivated means now apoptosis is blocked here. Number one. Other is inhibition of pro-apoptotic transcription. This AKT is unable to activate the transcription factors of the pro-apoptotic genes. Pro-apoptosis genes are this BIM and PUMA. These are like inducing apoptosis. These are inducing cell death. These transcription is inactivated. That means these genes are itself not synthesized. All these genes are not transcribed. These are not synthesized. So it will, like, there's no apoptosis happening. So cell survival will increase. So this activated AKT, it is promoting the cell survival by blocking the apoptosis, by blocking the genes responsible for apoptosis. Ultimate final outcome is cell survival. By blocking the apoptosis, by blocking cell death, the cell will survive. Final outcome is cell survival. This is about the AKT. Coming to the step 4, it begins with mTOR here. mTOR means, the full form of mTOR is mechanistic mechanistic target of rapamycin. Mechanistic target of rapamycin. This mTOR, activated mTOR, it has two functions, like it has two complexes. mTOR C1 complex, this is the mTOR C1 complex, and this is the mTOR C2 complex. The mTOR C1 complex is involved with regulating the protein synthesis and cell growth. Protein synthesis, cell growth are regulated by C1. The C2 responsible for regulating the cytoskeleton and activates the AKT. This C2 will in turn activate the AKT. Now, this AKT, this ultimate the function of this mTOR is to increase protein synthesis, increase the cell mass and proliferation. It will increase protein synthesis. By increasing protein synthesis, the cell mass, the cell growth is increased, and also cell is ready to divide, proliferate. Now, this mTOR, I'm saying we began the story with activated mTOR, but this mTOR fate, it depends upon TSC, depends upon molecule called TSC1 or 2. Tuberous sclerosis complex. The full form of TSC is tuberous sclerosis complex. Tuberous sclerosis complex. This tuberous sclerosis complex, it will have a negative impact. It will inhibit mTOR. Tuberous sclerosis complex 1 and 2 will inhibit the mTOR. Now, whenever AKT is active, this activated AKT will destroy, will phosphorylate, it will add phosphate and inactivate the TSC complexes, tuberous sclerosis complexes. Whenever the tuberous sclerosis complexes are inactive, that means mTOR, the break, this negative impact is lost. Therefore, the mTOR pathway is activated. So, this activated AKT will phosphorylate the tuberous sclerosis complex. Tuberous sclerosis is shut down. That means the negative break on this mTOR is lost. When the break is lost, it gets activated. And this mTOR again, it will via C2, it will again increase the AKT. AKT again, it has like a loop. It's like a vicious cycle. Again, AKT will suppress tuberous sclerosis complex, and again the mTOR gets activated. More active mTOR, more AKT. More AKT means again by suppressing the tuberous sclerosis complex is more mTOR. This is like a vicious cycle. The ultimate thing is to increase protein synthesis, cell growth, that is mass and proliferation. This is about the step 4, that is the mTOR pathway. Coming to the regulation of these cycles, this cycles has to be regulated. Here the breaks are the PTEN, that is, PTEN is a tumor suppressor gene. It will have a break on PI3K pathway. On PI3K molecule itself, it has a break, and it will ensure that the cell cycle is not proliferating uncontrollably. So it keeps the cell cycle under check. Here, it is cell cycle provision occurs. This is kept under check by this PTEN. Whenever there is a loss of PTEN because of mutation, this PI3K will be overly activated. There's no break on the PI3K, so overactivation will cause uncontrolled cell proliferation, ultimately leading to cancer. Uncontrolled cell proliferation will lead to cancer. And here also at the mTOR level, there is an APMK, which acts like a brake on mTOR. This APMK is activated under low energy states. Whenever the energy stores are low, whenever the energy is low, the APMK will get activated and it will break this, it will act as a brake on mTOR. So it will not progress further into the cell proliferation. So ultimate aim is whenever there is low energy states, in low energy state, APMK will be more, APMK will inhibit mTOR. The final output is to decrease protein synthesis, to decrease the cell growth, to decrease the cell division. Whenever there is low energy, whenever there is low energy, the cell growth will be low. It has to shut down the cell growth and proliferation. That is the ultimate target. So this is about the second branch of the cascade PI3K AKT mTOR pathway also we have discussed. Now coming to the clinical correlation here, whenever as I said, whenever there is a loss of PTEN, PTEN is a tumor suppressor gene. Whenever there is loss of PTEN because of mutations, the break on the PI3K is lost. So this loss of PTEN will result in certain syndromes called Cowden syndrome. Cowden syndrome is like a triad of breast cancer, thyroid cancer, endometrial cancer, or it can cause simply endometrial cancer and a glioblastoma. At the PI3K step, activating mutation of the PI3K will lead to breast cancer, colon cancer, ovarian cancers. Breast, colon, ovarian cancers are caused by mutations, activity mutations of PI3K. Here at the AKT step, overactivation of the AKT will result in many solid tumors because of overactivation of the AKT. At this mTOR activation will cause cancers, many cancers and tuberous sclerosis also. This is about the clinical correlation of the signaling pathway of the receptor tyrosine kinase. So this is about the receptor tyrosine kinase. In our next video, we'll continue with the non-receptor tyrosine kinase. If you find this video useful, please like, share and subscribe to our channel. Thank you.